That's what I have.
Okay, great.
Marley, for the video transect, do you want to push in a little bit on the Herc HD past the
Yeah.
nip? Yeah, you're in the box, so we'll have to wait for the ship to
Yeah, the
catch up.
ship's been moving. So.
Yeah, sounds good. I haven't tried messing around with the auto XY, but up to you if you want to fly it.
See that crab to the left holding on to the sponge?
Yep. He's going to make it on transect. We'll be able to count him.
So is that white fluff below this sponge coral?
Yes, that's that Clavularia, looks like. Yeah.
We have,
Yeah, we have.
yeah, sorry. Yeah.
Very pretty.
And there's a coral right there. Come back out to the mud.
Sorry. Can you say that again, Finn? It's hard to hear you.
Oh, I just said back out to the mud.
Get to the target. Okay. I mean, if we go even straight to the target, we get a bit more upslope, and we'll probably see more sponges and stuff for them.
Sounds good.
Yeah, we can do it on the next one. Sure. Yeah.
And now we even lose the sea pens.
I know,
Yeah.
this seems like very underutilized habitat.
Also, a very different colored substrate, or more sandy compared to other areas where it was very green, or could be the lights.
Well, there's something different because we don't have the sea pens. So
Yeah.
they don't like it for some reason. Black-gill rockfish. There's more sea pens in this mixed area than there was on the mud.
True.
Strange. Yeah, I'm gonna do 315. Is that all right? Yep.
And you can see the thorny head up in the right is a short spine because you don't see the long spine in the dorsal fin.
There's the fin of the caudal fin of a fish
Yep.
under a dead
Yep.
sponge.
Trying to hide from us.
I can hold off, yeah.
So,
Oh.
what are the tube color type of? Oh, to hit that. Come along with us. What are the tube sponges called? That one that just came with us with the header coning?
The tube sponges, I'm just calling them tube sponges.
Okay. Good enough.
That's why we collect them, to try to get an ID.
Right.
There's an asbestopluma, the predatory sponge. That stick.
Oh yeah, now I see it. The left?
Those
Yeah.
ones are Paragorgia,
Oh,
but the far left there was one, yes.
do you want me to do 310 instead, or are you guys fine with that? Okay.
Fish is swimming backwards.
As its backup. Meep, meep,
Yep.
meep.
Yeah, we still have 100
There-
meters to go in this transect.
Roger that. Lisa, you can provide all the sound effects.
What I can be good for. What I can excel at.
Naming things and sound effects. There you
Well, not, yeah, okay. Not the proper name. We'll leave that to you.
go. I don't know, the Farrah faucet, that was good.
Yeah. Proper names are overrated.
You never know. Could people could adopt it.
You know, it's just, you say it enough times, just like when we saw the benthic siphonophores, and we call them pineapples, other people call them other names, so if that's what the group calls it, that's what the group calls it.
Yep. And before you
Yeah,
know
that
it,
is true.
it'll be used at a scientific conference, and it's just how it goes.
And the density of squirrel tails on this mud was,
Can you lateral a little?
Ooh,
Sorry?
Do you lateral a
What's
little
up?
to the north?
Yeah. Oh, yeah, I think
So-
I think he's fine. We're all good.
what's that? We're just passing it by. It looks like tiger striped. Well, you can't stop and look. It's on transit, so
yeah, I saw that was on the side of the rock. I don't know,
Yeah.
something encrusting.
okay.
Couldn't see
Just
it well enough.
just had a very interesting pattern.
It did.
Yeah.
You might
It
want to
was
hold
the
off
bars
for
of the
a while.
jail cell.
Yeah.
Otherwise, everyone's gonna yank you around.
Yeah, I meant to say zebra striped rather than tiger. The other animal. Lots of sponges here.
I think we're at the end of our leash.
Yeah.
Grenadier coming up.
Mizumia.
That would be the correct genus.
Yep,
Looks like he
because of the
got
black
bit or
fin.
something and
Yeah, it's like 4 inches.
two-thirds of the way down the tail. It looks like there was a bite or some kind of injury.
Oh, yeah.
We're coming back to a big sponge area now. What we had a few hours ago.
Rockfish there with the white head.
Yes, that's a black gill.
Oh,
It's called a black
Oh,
tail because it has a white head.
yeah.
Oh, whoa. That's a big one.
Oh, there was another one right there in the left-hand corner, but could barely see it. It was very large. Rockfish.
Did you mean the right-hand corner?
Yeah.
Right hand, yes, sorry.
Okay.
For monitors.
Yeah. Did you see that, Tom?
Yeah, that was a short spine thorny head.
I like when they have their pectoral fins out like that.
Yeah.
Yep. Cool things are when the skates are using their fins to walk instead of swim.
Oh, yeah.
And you just walk on the bottom.
Like those batfish. I've seen that.
Yep. Just kind of like that.
Yeah.
Never seen that. Would be cool to see that.
Well, perhaps we'll see one in a while. I haven't seen a skate in a while, though.
Yeah.
So these white sticks up here are that predatory sponge, the Asbestipluma. Or we'll say Asbestipluma-like, because Abby was saying there's another species it could be, or another genus.
Another genus of predatory sponges or carnivorous sponges.
Yes.
Yeah.
Cladorhiza or whatever. There's a hagfish. Woo.
A couple of them.
Yep.
They don't get the credit they deserve.
I know. There's something adorable about them to me. The way they search with their heads. I don't know why.
Yeah. I've never heard
I
of them
think
called
we have
adorable before.
170. We've got 150. Yeah. Yeah, I don't know why. I find lizards adorable too.
Well,
Lizards are
lizards,
fun.
yes.
What?
We agree with you on the lizards being adorable,
Oh.
but hagfish, I disagree.
I've never gotten slimed by one though, so maybe
Yeah.
that would change my opinion.
Exactly.
All right, so here's my hagfish story for you, because I don't worry about the slime, I worry about the jaws.
Oh yeah?
I was trawling, or we had done a trawl and we were separating the fish into buckets. And just because, kind of like this, you're up in the middle of the night doing whatever, and I grabbed a hagfish and I'm holding onto it, and next to it was this fish called a slickhead. They were in the next bucket.
Thanks.
So I like stuck the hagfish's head down and said, look, It's a slickhead, and he just opened up his mouth and took his rasping jaw, went straight down the side of the fish and opened it straight up.
Oh, wow.
And I'm like, okay, I'm going to leave you alone.
All right, maybe adorable was the wrong word.
But even without jaws, they can use that rasping
That's so
part of their mouth
interesting.
to just completely open it up.
Razor, yeah.
Yeah, and it's very scary looking. But if you look up at their jaws,
Yeah.
Yeah.
the spiky teeth, it's not adorable.
I had a friend who grew up in Florida, and his brother got bit by a cookie cutter shark, the ones that like
Mm.
sit up and
Mhm.
down
Yep.
and just come and twist. That sounded really not fun.
Yeah, that would suck.
Yeah.
The Faunivore, he just went by. Left hand, service 3.
Which left? No, never mind.
Yeah, and you should call me out on that because I'm dyslexic that way.
I don't think it would really, I do the same thing. No, I have to think before I say it because if I just shout it out, I only have a 50% chance.
Yeah,
It's getting really popular.
that's basically what I'm doing.
So that means you're right 50% of the time. That's good.
I'll take it.
There's a tanner crab.
And how much further on transects?
Yeah, we have about 20 meters left,
Okay.
so almost there. Gonna bring us right to that target, pretty much.
Science, it looks like Herc's going to be a little bit off of the AC target. We can lateral over there.
Yeah, I mean, it's your
There's
20
nothing
meters
magic
away.
about the spots.
Yeah,
Yeah, that's
So-
okay.
Oh,
Yeah.
cool.
Sounds great.
I wish there was magic in each of the spots.
But
Your
science,
pot of gold.
we trusted you. No, I'm sure.
So, okay, now I know the first mistake.
Oh, Abby's on. Is this fun for you, Abby? All these sponges?
She just said it was incredible in the chat.
Yeah.
So
Oh,
she's going to get on and you guys are going to move 5 meters and collect everything.
But if you see something, Abby, because we'll be ending the transect soon,
yeah, we're about there.
that you think we should collect, let us know.
right, so that's good. We are. Should be about 200 meters.
Roger that. End of transect.
All right, thank you.
That's the other kind of ground fault. Dragging the tail down slope.
Yeah. If you want, you can always.
With 20 meters to go. I wouldn't change the heading.
I think the transect
Yeah,
just
the
ended.
transect's
correct?
over.
Yeah.
So if you want to go, so we're going downslope right now.
Yeah.
Starting to go downslope. So I don't know if there's any preference to, I mean, there's still sponges here, so if there's anything.
Yeah,
Yeah,
I think we,
Sorry, I'll come down.
let's just look around. Since we only got 13 minutes or so till
Yeah.
watch change. We just kind of look around here, go slow, see if there's something we want to collect.
The ship's up.
Yeah,
So
and we should watch the science chat to see if Abby has any instructions.
She's asking us to find a Thenia, but I don't see one currently.
also, Kim, I'm going to reset you. You're off autos.
So Abby, you want a yellow one, like the one coming up to the left? If we could stop and try this yellow sponge just on the bottom to sample.
The plate looking one?
Yes. She would like a zoom on it, and she could tell us.
Is that that one?
Yeah, that one right there. Yeah.
The one right in the foreground?
Correct.
Yeah.
This really thick one. She wants to check and see if it looks like a certain species, and she'll decide on whether we collect it or not.
Marley, Marley, you wanna push it on Argus cam there?
Sure.
Since we're sitting down. A little sponge field.
Yeah, it's a cool shot.
Digging that shot, Marley. Thanks.
Yeah, me too.
I'm gonna try to zoom on that. I don't feel like I'm very stable.
Yeah. Marley, you wanna zoom on Herc there?
Oh, yeah.
What you think, Abby? Would you like to sample this one?
It's a cool sponge.
It is
Plenty,
nice. Look at all the. It's got those little furrows in it. Kind of cool.
Trying to convince Abby.
What? No. Yeah. Yeah.
Sponge like it, though.
Yes, she would like to sample it. So, any
Cool.
particular place, Abby? It looks different, but she's not sure.
No
So,
particular
she
place.
said no particular place, just any old snippet will do.
Roger. snip, snip.
Haircut.
Wait, I thought Lisa was doing the sound effects. She gets to do the snip snip.
So, oh, Roger that. Yeah. Before Bob gets in here, takes all of our fun. He's listening. I hope you heard that.
Mary, this is 070. Okay.
You want to come wide there, Marley? On Herc. So science, we have a couple options here.
Okay.
We can take a snip, take a slurp, or take a snip, throw us in the starboard bile.
How about we do a slurp? Save some bio boxes.
Roger. Slurp, save bile boxes.
That works for me.
And this is sample 70, 70.
Kim, would you mind racking back a bit there,
Roger.
please?
Yep.
It's been
And can we throw on the light if we get a chance? Oops. Thanks, Kim. All right, I'm ready. Ready for a flush. Roger. Yeah, we can see how it studies. Roger that. Oh, I can also do a cookie cutter out of it. 30 should be good, I think. I think that could be good, Kim. And if we pan over a little bit, then I don't need a full zoom on this guy. Riley, gonna do a slight push there on the Herc HD. That's grand. Almost inclined to see how far we are on the hose there. Ah, it's still good. All right, taking a little bit of a bite. I need a little more jam on that suction pump there.
Are you putting in the flush?
Oh, good point. Sorry. I
Sorry.
don't want that. Oh, that's a great catch.
Good catch. That was awesome.
yeah. Oh, keep on going.
You want to zero the suction there, Kim?
No suction.
All right, so let's do 3.
3 is
Uh-oh,
good.
boss man. Yeah,
Uh,
go ahead.
Wow.
All right, go ahead. Go ahead. Yep, please. Now, give this guy a little shake.
Come on, go in.
All right. Riley, you want to push in a bit more there, please?
It almost looks like a fish head sticking out. going in?
Let's
Yeah.
see.
See how this guy settles out.
Oh, it went
No,
in. Yay.
I didn't see it go in.
No,
It,
it
it's
went in
not
the
at the
front. I
there's
entrance.
don't.
the sound
It
effects.
went in
on
the
it.
Yeah.
front. Yeah.
Oh, here we
Oh,
go.
there we go.
It emerges.
We can't see the jars, so I
Oh.
have no idea if
Yeah.
it's in or not.
Oh, well, we got
That's why we're
about
beating you to it.
half a fist size of it in there.
Yeah, it looks,
Fist size, maybe. That's good.
That's good.
All right, come wide there,
I'll
Marley.
believe you.
And in which jar is it?
It's in jar number 3.
Thank you.
Thanks, Kim.
Well done.
All good.
yeah, and so we're getting ready for our watch change up here, just so you guys know.
Yeah, boss man's here.
Boss man. Boss man, boss woman.
We're up for a change as well.
Yeah.
It's brilliant to us,
I don't know.
as usual.
Thanks,
It
guys.
was.
That was good.
Good job, everybody.
Yeah, thanks for joining us.
Yeah, very
we
good.
appreciate it.
I'll see you back again at 4 to 8 this afternoon.
All right, sounds good. See y'all then.
All right.
Okay, thanks,
Thank
bye.
you.
Adios. So scientists, I'm signing off now if someone wants to sign on.
So I think it's who's coming on after us? I'll stay in to see who comes on. Not sure. I'll check it out. So hey Abby, how you doing? Good. So you're on? Okay. Yes, you did a good job. I'm not hearing the echo anymore. So as you saw, we just finished the transects, and that was transect 6. We did 3 on our watch. So I guess you can have in this field of sponges, if you want to collect, a good place to do so. Any questions before I sign off? Yes. I didn't hear that last part. It's a little bit of echoey. Yes. And I think I can hear you probably on the Nautilus channel, but you need to turn on the SPL channel. It's the red bar in
Got
the
it.
link.
Yep.
Okay, great. Then I will sign off, and I have, yes. Hi. I can hear you really well. Let's wait. I'm going to ask Tom if he can hear you. Yeah. Yeah, we can hear you really well. And then just turn on the SPL and then I'm sure it's great. No, I can hear you really well.
Okay, Sam, what's the deal? I heard something about going up in the water column.
I haven't heard that yet. The team has been running transects. I know we just completed a 200-meter transect, and now I think there's some question whether we want to keep looking around here or continue on to our next waypoint. So, science.
This is a long ways?
Do we have science yet? This is the what, Bob?
Not yet.
Not yet.
There are 4 jars left, so 4, 5, 6, and 7. And you've got the forward bio box wide open, and you've got the 2 small starboard component bio boxes open as well.
Looks like 1 Niskin fire?
That's correct. Two Niskins.
2 Niskins. 1, 2, 3, 4. Yeah.
So, Bob, we have targets now.
Wow.
I know. Not sure how it happened, but happy to have them.
Somebody bothered to make a target file. Yep.
Cool.
That's very handy.
Sure is.
We got underlays over here. I'd never look over there again.
Okay,
Well,
what do you think about this Niskin die? Yep, yep. Yep. Yeah, I think that would be great. Yep. Yep.
Maybe I'll put that on my Christmas wish list.
Targets on the
Underlays.
nav tape.
Targets, they've always worked.
Yeah.
That's just, somebody just has to make the target file.
Roger.
You make one for that, right? Is that just a comma delimited thing?
Great question. Haven't gotten there
Thanks,
yet.
Lisa. Thank you.
Good morning.
Morning, Nautilus.
Good morning.
Good morning, Nautilus.
Good morning. Science, we understand that we've just completed a transect, and the question now is, do we want to stop and look around here a little more or continue on to AD?
I think we'd like to stop and look around a little more. There's actually 2 things that we're just talking about on the watch swap over is taking a Niskin here, and then also right where we are, there's a sponge in the field of view at the moment that we'd is a potential sampling target that we'd like to have a little zoom on.
Perfect. Which sponge?
So it looks so it's off center, slightly to the left. Oh, hang on.
Set 3.
Frozen screen.
For Steve,
It's
I'm going to
small,
put it straighter.
It's round. It looks like a little button almost.
I'm
With
circling it for them.
You zoom in
darker
now?
brown fringe.
Zoom in.
Oh, out of frame?
Oh, hold on. We're going to use some telestrator here too.
Yep, I got the telestrator up.
On channel 3. And you should be able to see it, ladies, in just a minute.
Yeah, this
Seconds even. You sure it's that one, Steve?
Yeah, I'm pretty sure it's that one.
Yeah?
Okay.
Yes. Yeah,
That's it?
I'm not positive it's it, but if it is,
Okay, you want to zoom in on that, Al?
I'd love to.
I knew that.
Wow, look at that zoom. Isn't that something?
That is.
It's the best zoom you're going to see.
wow.
Got a little wiggle going on though.
Makes it exciting. Induces drama.
Right.
Yeah,
You
so
feel-
I think there's actually two of them there.
up.
And yeah. And the thing is that we've tried to collect the sponge before, and it's actually quite tricky because it's got these thick, long spicules around the edge. So there's two of them, one right in the center, and then one kind of at like just slightly up and to the right of it. And it's got this white center and brown spicules all around, and it's previously been pretty hard to collect. We would like to collect a whole specimen, but it has been a challenge. They
Okay.
tend to sort of fall apart.
So,
Yeah.
can you zoom out a little bit, Al?
There's a little bit.
Can we turn on the lasers? Yeah. Huh. So it sort of looks like it would clog up in the hose probably.
Yeah, it's got really long kind of spiny spicules. I think it should be a sinker if you can sort of scrape it.
Yeah.
But yeah, like I said before, it's been a tricky one.
So it's got to go in the side compartment.
Doesn't have to.
Can
If you want to put it in the forward, we can, but you know, we would just be able to add a sponge or sorry, a coral later to keep it distinct.
Yeah. Well, I'll pick it up and then we'll see what it looks like. Whether, yeah. Okay, zoom out, Al.
You want to do that collection first, or you want to do the Niskin first?
Talking to me?
Anyone.
Your choice, Abby.
Yeah. It doesn't. No preference.
All right, we'll do the Niskin first.
Okay.
Everything's kind of settled out here.
So Abby, what is the story on this sponge? Why is it of such interest?
So this species seems to be pretty common. We have a tentative ID. Initially, it was identified as the muricata, but sponge taxonomist Henry Weiswig then kind of looked at it again, and the muricata is more of an Atlantic species, so it's unlikely that we're looking at the same species here. So there's some just kind of taxonomic uncertainty over what species it actually is, and that combined with the fact that it's actually quite difficult to get an intact or mostly intact specimen up to the surface has made it a bit of a challenge. And as it's also one of those sponges that is morphologically distinct enough that we can ID it from imagery if we can get a name on it without taking a sample. So if we can get a solid ID on it, then that will mean that in the future, all of these visual surveys that we're doing, we're just going to be able to like know this is what this is and be confident with that identification.
Steve, we're doing number 3?
Number 3.
Okay.
All good?
Yep.
Can you come wide on Argus? Thank you.
Do you want to come all the way out?
that's good.
Could I just get a check on which sample number the Niskin was, please?
Yeah, that's 071.
Thank you.
Steve, is that at the same site as the sample 070?
Yeah, we didn't actually move.
Roger, thanks.
Hey, you want to zoom in now? We have, so that one, right?
Yep.
Right?
So there's actually there's three I can see now. So there's the one, yeah, looking like
oh, and one up
right in the center.
above it. Yeah.
There's one, yeah, one just kind of above an ophioloid, like above that little brittle star. That's another one there too.
You got a preference?
No preference.
Right.
Maybe the one to the right looks like it's a bit more separate from, you
Yeah,
know,
that was my
other things
preference,
going around
I
it.
think.
Yeah, yeah, that
They can
sounds
come
good.
in from the side easier than trying to pluck it out of the middle there.
Yeah.
Okay.
Challenge is on.
Yeah, to get the pan itself to work.
It's an
All right,
interesting.
well, you can zoom all the way out there. Or did we get enough shots? Are we happy?
Yeah.
Shots?
Yeah,
okay.
we were happy.
This is Di. I'm just saying a testing 1, 2, 3 to see if others out there can hear me since I had audio trouble yesterday. Thanks.
Di, you sound fantastic.
Out to get me today.
Thanks, Al. Good to hear your voice.
It was cold and chilly, wasn't it? Condensation dripping down the outside of the glass.
Little inside joke. Sorry. Yes.
Someday we'll be on land again.
Okay, I'll zoom in.
Going in. Well, when the claws get there, you see just how small it is.
Yeah.
Oh, this is all like on a substrate here of some sort. There's a lot of keyboard clacking going on.
Yeah, somebody
clacking away back there.
Liz, I think it's you.
Huh. Yeah, that's.
It's giving me a thought though. If I see it on a smaller rock, that might be
I
an
think
easier.
I might be able to separate the rock here, maybe.
That would be ideal.
Or not.
Abby, is this another of the same sponge on the bottom right?
Yep. Nice.
What you're thinking on picking that whole piece of rock up?
Is it small enough to go in the one of the front bio boxes?
Oh. Zoom in just slightly there. Huh? So it's 10 centimeters wide by. Well, is this other part all solid here?
The left piece might be smaller now.
Yeah, actually, huh?
A lot smaller. Jeez.
Yeah. Well, how about this other one then?
Either is fine.
All right. Let me zoom in some more. You kill the lasers?
Yep.
Oh, it's two pieces.
Smaller.
Even smaller. Perfect.
So it's underneath that on the left, isn't it there? Yeah.
Yeah.
I think that'll be the challenge, is just once it comes up, like spotting where it is on the rock.
Yeah. Let me try and scoot it over.
You've got a few other sponges. Yeah.
Can we do a little rotisserie? Just to make sure we see which side of the rock it's on.
Zoom in some more out. Oh, there's its, what do you call that thing? Opening?
The osculum, I think it was actually, I think it was, if you shimmy it around a little bit more, I thought it was under to the right here, so kind of right in the middle.
Yeah.
Because it should have this like fringe of brown spicules. So I think it's actually.
I think you're right, Bob.